Okay, moving on to a bit more on the genetic side now, on the topic of evo-devo, so evolutionary development, how embryos, how development at the embryonic stage leads to evolution in a lot of species and in a lot of ways very fast, rapid evolution. So, developmental biology studies, sorry, developmental biology studies how an organism's structure changes over time. Prior to Darwin, the term evolution had been essentially a synonym for development, but the Darwinian revolution changed the term's meaning, and they became distinct. Now we're learning that evolution and development are more closely related than we once thought, hence we put them together in the school of Evo-Devo. There were originally two theories for embryonic development: the preformation theory, which postulated that all structural components were present in the sperm. This is a rejected theory because it wasn't true. If you look at this image here, it's kind of what the preformationists thought. They thought that at the kind of acrosome head of the spermatozoa, that you basically had a fully formed well, not a fully formed, but a pretty decently formed human with all their kind of structural components, and then they fertilized the egg that gave them the resources they need to grow. We now know that's not true. The other theory, which we now accept, is called epigenesis, which postulates that organs and tissues form anew in each embryo. So you have the spermatozoa fertilizing the egg, they share their versions of chromosomes 1 to 23 together to make a diploid cell. The diploid cell divides to form a four-celled tetrad in the zygote, continues to divide, you get the morula. Then the morula continues to develop germ layers. You get stuff like plastulation to form gastrula, and then eventually you get a digestive tract, you get all the limbs, you get all the tissues, you get a fully developed embryo. The Darwinian view of evolution was then adapted to the modern synthesis that we saw evolution in scales of changes in allele frequency over time, rather than just observation of adaptations being selected through natural selection. The science of Evo-Devo in no way implies preformation to be true. It simply asserts that changes to the homeobox genes responsible for structural development, so the genes responsible for all this, in the embryo, these homeobox genes, changes in them result in profound phenotypic changes in a single generation. So large leaps in evolution. You can go from, I don't know, you can go from having, say, two eyes to having one eye if you kind of get structural development mutations in homeobox genes causing cyclopsism, or whatever it's called. Or one of them that we're going to talk about here, you can get extra limbs, or say you get mutations in the antennapedia gene, and let's just say by random translocation of the gene responsible for leg development, the antennapedia gene, say that randomly gets reintegrated into the antennae homeobox region, and then you'll get flies with legs growing out of their faces, where antennae should be. And when you think about it, if that were to be an advantageous adaptation and it were naturally selected, and then the organism with the legs in front of its face is able to reproduce more and increase the frequency of those alleles in the next generation, that's essentially rapid evolution. And you've gone from having 6 legs in the case of flies to having 8 legs and 2 in the face. So And then those structures can obviously be adapted to better serve their function through natural selection. So there are three main steps to embryonic development: the cleavage step, the gastrulation step, and organogenesis. So cleavage occurs immediately after fertilization, where a zygote is partitioned into smaller cells called blastomeres. So when the zygote divides to form a structure between 2 and 8 cells large. During the cleavage stage, cell cycle arrest stages T1 and T2 are lost to allow cells to divide faster. So this is kind of an image of the cleavage stage up here. Notice how the zygote itself, as it's dividing, it's not becoming larger. Like, it's the difference between the first one on the top left labeled A and then the one labeled C. A is a single cell, C is a tetrad made of 4 cells. The tetrad isn't 4 times bigger than A, it's actually the exact same size. It's basically just A divided by 4, so each of the cells in C is 4 times smaller than the original cell in A, and the cells just get very, very small for the embryonic development stage. So for gastrulation, this refers to the process of plastopore invagination in the blastula, which is when this cleavage stage gets to, I forget the exact number. The morula is 32 cells, and then the morula develops more germ layers to form the blastula, which we show here on the left. There is a pore in the blastula called the blastopore. This pore invaginates, as you can see here on the image on the right, this kind of purple structure. That's the pore kind of invaginating to kind of form a digestive tract. So imagine you just get a grape or something and you get a pin, you stick a pin through it, you take the pin out, and then the grape has a hole going down the middle of it. That's kind of how it's working to form the digestive tract. In protostomes, the blastopore becomes the mouth first, and in deuterostomes, like humans, the blastopore actually becomes the anus region. So, moving on, the process of organogenesis refers to the process of the 3 germ layers giving rise to organ systems. So, in the embryo, once it kind of divides to the gastrula, you can see here the blue cells labeled ectoderm, green cells endoderm. I don't think mesoderm are labeled here. I think they develop slightly after the gastrula. But these germ layers give rise to the different kind of structures that are going to be present in the more developed embryo and in humans. So the endoderm layer gives rise to the lungs, liver, stomach, intestines. The ectoderm gives rise to the brain, skin, and the nervous and endocrine systems. And then the mesoderm layer, which is generally rarer in the animal kingdom, only found in triploblasts. That the mesoderm gives rise to the skeletal muscles, the heart, and the bones. So these developing cells move where they need to go through genetic signals. We're running out of time, so I think we're going to continue this in the next one.